In 1961, scientists Marshall Nirenberg and Johann Mathieu sought to solve the genetic code. With the use of an enzyme called polynucleotide phosphorylase, they were able to determine which RNAs specified which amino acids. Polynucleotide phosphorylase, unlike RNA polymerase, is able to randomly link available RNA nucleotides and does not require a template. These randomly linked nucleotides also created homopolymers, or RNA molecules that only contain a single type of nucleotide. For example, a homopolymer could be uracil, 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 or poly-U. The poly-U RNAs were then placed within tubes consisting of translation components. What they found after the mixture was that uracil specified the amino acid phenylalanine. This process was then repeated for adenine, cytosine, and guanine. However, polyguanine RNAs could not be interpreted. To further their findings, they decided to mix nucleotides. For instance, mixing adenine and cytosine resulted in 8 poly-AC RNAs, which coded for 6 different amino acids. It was noted that these amino acids were dependent on the ratio of nucleotides. A probability was created after several calculations using the observed ratios that determined the conclusion. In this instance, the amino acids with more C's than A's is more common than one with more A's than C's. In all, Nirenberg and Mathieu were able to discover base composition for genetic code, but were unable to decipher the orientation of the codon sequence. Issues were still prevalent with a non-random assortment of bases because many codons represented the same amino acid. Thus, 3 years later, Nirenberg and Leder sought to resolve these issues with the prior discovery of random copolymers. They used ribosome-bound tRNAs to reach a solution. For their experiment, they took short mRNA sequences and combined them with ribosomal tRNA with amino acids. The new ribosome-paired mRNAs with tRNAs were passed through a nitrocellulose filter, and only the unbound tRNAs passed through. With the leftover paired tRNAs that didn't pass through, they were able to record each codon that specified each amino acid, thus filling in the remaining gaps of the genetic code.